Hello, my name is William Lai, and I'm an assistant research professor in the Department of Computational Biology at Cornell University. Today I'll be talking about adversarial attacks for interpreting genomic neural networks. So my research program is based around this fundamental question, which is how do protein complexes organize themselves in the genome? We see in this cartoon from NHGRI that chromosomes are actually subdivided into nucleosomes, these small beads on a string that have genes arrayed on them. In between the gaps of the nucleosomes are where transcription factors bind, and we even have polymerase transcribing RNA here. And the overall arching question is not only how do protein complexes organize themselves in the genome, but in multicellular organisms like humans and mice and anything else that is a highly complex eukaryotic organism, where does the variety come from, given that every cell has essentially the exact same template of DNA behind it? After many decades of research, we know that cis-regulatory modules, more commonly known as enhancers, are likely the driving forces behind a lot of the cell type diversity that we see in multicellular organisms. These enhancers tend to make three-dimensional contacts on the genome in extraordinarily precise ways, where they recruit a lot of different transcription factors, sequence-specific DNAs, chromatin remodelers, enhancer RNAs, all come together to form this three-dimensional context in a very precise way, which kind of goes against the canonical way that we're taught in high school and college, which is that DNA is not a linear string of nucleotides. It's actually a very precise, compact combinatorial mechanism with a lot of different protein cofactors. Kind of confounding our ability to detect enhancers in cancer is the idea that DNA sequence becomes progressively mutated in cancer, which makes it especially challenging to track how enhancers change in a mixed cell population outside of doing single cell approaches, of which at this current stage we do not have single cell chip-based assays widely available. So a question that my group asked is, what if we inferred enhancers instead of using DNA sequence, which is generally the standard for enhancer prediction, but instead using the location of these exact balls on the string? So kind of supporting this concept is some of the image recognition technologies that have come out, namely deep neural networks. So we can think of, instead of predicting an MNIST numerical hand-drawn number, we can look at the actual way that the data, these ChIP-seq data, pile up on the genome in very precise ways, add enhancers, whether it's P300 or specific histone modifications, or signals of actual RNA transcription that form in very unique, precise ways, add enhancers. And instead of using these images to try to use them as a training data to predict where enhancers are, using only the vectors of the chromatin data, ignoring DNA sequence entirely from its actual training. So the first thing we need to do in order to test this is actually to generate training data. We start using well-characterized ENCODE cell lines, and we start with intersecting ATAC-seq and STAR-seq to create stringent and lenient enhancers. So ATAC-seq is a well-known gold standard assay for detecting a measure of accessible chromatin in the genome. It's considered to be generally biochemically active wherever you find an ATAC-seq peak in the genome. STAR-seq is a pseudo in vivo in vitro assay which measures the DNA's potential to act as an enhancer. Critically, traditionally STAR-seq assays are in a no chromatin context, so that there are no native nucleosomes or proteins in the substrate other than the local whole cell extract that's used as part of the STAR-seq assay. So by intersecting these two at different p-value thresholds, we can create a list of stringent and lenient enhancers that we can use as the training data for building a model. So, just some orthogonal validation. What is in our training data? Does it make sense? If we pile up tags from downloaded ENCODE datasets across our 4 cell lines, A549, HepG2, K562, and MCF7, we see strong enrichment for K27 acetylation and P300, and we see strong de-enrichment for K27 trimethylation and K9 trimethylation, which makes sense that our training set is enriched for markers of enhancers and depleted for markers of repression, and because we selected these enhancers independent of these ChIP-seq datasets, we are trying to avoid any circular definitions of enhancers. So we can take a subset of the data, so K27 acetylation, K4 trimethylation, CTCF, POL2, and P300 as our training data, and we can actually use just the distribution of the aligned sequence tags at all of our predicted lenient and stringent enhancers minus some holdout cross-validation data and attempt to train a model. And the model architecture we went with after some substantial hyperparameter testing was the very popular CNN to LSTM to DNN network. So the question is, how did we do? So if we then look at our holdout data, in this case it's chromosome 17 we held out from our training for some of these models, we see almost a perfect match against our training data in terms of the histone modifications that are enriched inside our holdout prediction regions. And we see very nicely we have an AURC of 0.88, AUPRC of 0.25. So our network seems to be relatively sensitive and precise to identifying enhancers using only the chromatin data tracks. But the kind of the overarching question is, once we have these networks, it's what did these networks actually learn, and whether or not anything it learned was actually biologically relevant. Because it's one thing to have a highly accurate model, it's another thing to ask the question of whether or not this model learned any biology, or if it's functioning purely as some modeling some sort of artifact within the data. So we wanted to take a bit of an orthogonal approach to our explainable AI approach, which was actually starting with particle swarm optimization, with this idea that we could initialize randomized vectors of chromatin data and adversarially attack our network. Since at the end of the day this is a binary classification problem, we can optimize our randomly initialized particles towards which are the most by random chance looking like an enhancer, and then through gravity pulling social weights of the particles, communicate that information to all the other particles and slowly converge on something that the network thinks across all of the particles in the system looks like an enhancer. So this is kind of what the raw data looks like. I'll walk you through it real quick, which is we have 250 particles here. Every single row here represents a different particle. They're initialized with a randomized ChIP-seq data. So this is 500 columns long, representing 1 KB windows at 50 base pair resolution. And we have CTCF, H3K4 trimethylation, H3K27 acetylation, P300, and Pol2. And you can see by the random smattering of the data here that the data was just completely randomly with a 0 to 1 random initialization. This is our in silico ChIP-seq data, and all the particle swarm plots I show you are actually ranked from best particle to worst particle. And the question is, what happens if we iterate across 20 epochs of the particle swarm optimization? What I'll show you on the right is actually the sum of each of the data sets. It makes interpretation just a tad easier to see what the general trends are across the particle swarm optimization. And you see that relatively quickly you converge to some striated patterns on the left, and when we sum that up on the right, we see very quickly that the data sets that are primarily enriched are K27 acetylation, followed by CTCF, followed by P300 and Pol2, with H3K4 trimethylation apparently not contributing a lot of information into this particular run of particle swarm optimization. Now we can actually take the trained network and we can run saliency or activation mapping on it and examine specifically from a gradient perspective which of these patterns actually made an impact in terms of the inference of the network at the end. And what's really cool to see here is that completely expectedly the K27 acetylation is blowing up all across its entire data set. But what was really interesting to see is that the CTCF is actually showing positional information in terms of a preference on the far left and far right. So we're actually seeing positional preferences for network activation in the CTCF, which kind of matches the biology of the system, wherein we know that the different proteins and the different histone modifications should have different positional preferences at an active enhancer that would be required for that enhancer to function properly. What's really nice about particle swarm optimization is that you can run it multiple times. You generally get out similar profiles. You can see across 4 different replicates particle swarm optimization. In every single case, K27 acetylation was the dominant data set in terms of providing predictive power towards a predicted enhancer in our trained model. But the other orthogonal data sets were slightly unique and likely represent different combinatorial possible patterns of enhancers. So, the overall arching question is, well, now that we know particle swarm optimization was robust and capable of identifying enhancers completely de novo from random data given a trained network, how well does this system actually scale? So instead, we can actually go to the ENCODE data, download, every single unique K562 ChIP-seq data set, and train a network on 329 K562 ChIP-seq data sets, and repeat particle swarm optimization. And this is essentially the network we get, trained across 20 epochs. And you see, again, this is the data summed up now across 329 data sets to a single column, just because actually visualizing the entire 33,000 column network, it was not really a practical approach in terms of seeing the data. But we once again see very specific striations. We can pull that data out because every column represents a different data set. And we can see across these 20 epochs which data sets appeared to actually be important. And here is the data across 4 different iterations of PSO. And I just want to call your attention to some of the initial findings of this large network particle swarm optimization run, which is across all 4 iterations, the standard histone modifications associated with active enhancers that we had even used in training the smaller networks all appeared pretty consistently. We also saw components of the Pol2 complex appear in all 4 of the runs, slightly different components indicating it is a little different in terms of the weighting, which is expected by chance in the particle swarm, but again, a huge signal of active enhancers. And just in this one particular one, we also got GATA1, which is the canonical K562 sequence-specific transcription factor, that actually in the other 3 iterations of the PSO, GATA1 was nowhere near the top 20. So the conclusion we have from here is that the dimensionality in terms of how much biology may actually be encoded in this network may be enormous. And we're probably going to spend quite a bit of time diving into what particle swarm optimization, along with other explainable AI approaches like SHAP and LIME, can really give us in terms of teasing apart exactly what kind of protein organization is occurring at these networks. So just to summarize, I hope I've convinced you that you can use genomic data sets like ChIP-seq to identify enhancers completely in the absence of direct DNA sequence information. One of our forward goals looking downstream is to actually use data that was aligned using reference genome-free alignments in order to truly get away from a reference genome-based enhancer prediction calls. We've also seen that adversarial attacks using particle swarm optimization really can find combinatorial patterns that seem to be predictive as a whole of enhancers, but individually may not be by themselves predictive of enhancers. And that PSO on these large networks really serve as a really awesome low-cost way for hypothesis generation that we can test biochemically using various ChIP assays. So with that, I'd like to acknowledge and thank the members of my lab, the Core Genomics Core, the graduate student who is actually from University of Iowa, Jamil, who took point on huge amounts of this project, as well as my long-term collaborators and my funding sources. Thank you.